a scanner, it would, get malfunction and get torn to shreds, and it would probably get smashed on the side of the wall. So the way this is actually set up is that you have, so there's a kind of magnetically shielded room around the scanner, and there's a little sort of window in that shielding, and you have a big powerful projector, not unlike this projector, but usually probably 10 times more expensive, literally, that shines through a sort of telephoto lens onto a mirror made of non-magnetic material that the subject kind of has on top of their eyes, like about 45 degrees. So yeah, you can't have the display actually in the scanner, but you can shine it onto a mirror. Although actually, what you can also do is, if you want to spend even more money, you can have goggles which are entirely made out of optical fibers. So there's no metal inside them. So there's basically, these kind of long optical, I'm not sure exactly how they make them, but there's these long optical fibers going through a cable, and then the optical fibers all come and make a little kind of mini optic fiber TV screen in front of your eyes. And that's very good. You need that if you're going to do, say 3D stimuli, which actually you would probably be interested in. So yeah, the goggles are good for that. But you can't use a standard kind of display. They're kind of expensive, these setups, but once you've got them, you've got them.
Do they typically consume like most of the visual field?
No, you can. It's a little restricted. You should volunteer for a study in a
I did. I
sometime.
forgot what it was like.
Yeah, that's fine. It's kind of, you're kind of cramped in there, and
I think it's like 30 degrees.
Yeah, that sounds about right. You're sort of looking in this little kind of letterbox in front of your face, and yeah, you definitely, if you were interested in something like, really peripheral vision, that's a little bit harder to do. Although probably I wouldn't be too surprised if you can get goggles right now that actually do wrap around more. In fact, people even have, virtual reality headsets and stuff that they can use in scanners now. I mean, if you're willing to pay enough, you can actually get some pretty fancy displays. But the plain old simple trick of just having a good projector and a mirror actually works quite well. Although it has to be a front silvered mirror. If you look in a mirror, like a regular mirror, the silvering is at the back and there's glass in front of it, so you don't mess up the silvering. But that glass is a little bit reflective too, so if you kind of look really carefully, you can sort of see two of yourself. I don't know if you ever do that in a mirror. Anyway, but if you're looking at the mirror at 45 degrees, then you're really going to see two of yourself, or you'll see two of whatever you're looking at. And everything always comes in at 45 degrees in the scanner, so it needs. So you can't. If you're ever dealing with an MRI mirror, don't touch the front of the mirror because you'll mess it up, and they're very expensive. Okay, so this is an example of a kind of simple color space. Namely, this one-dimensional thing, although it goes in a ring of colors, and finding the tuning of particular voxels to positions on that color space, and then using that to figure out what color someone was seeing. So my hope is that this is a kind of less complicated version of what the Galan people do. So my hope is that this is a little bit kind of easier to get an intuition with. It's especially easy to get. I think you'll get more of an intuition. If you're finding this a bit confusing, I would recommend going to like Photoshop or something and actually playing around a little bit with color sliders, and you'll get a feel for how you can make different colors along this continuous space. Okay, so let's have a look at some of the Galant stuff. I think what we'll probably do is before going into just too much detail about how it actually works, maybe let's show a couple of videos. Now these videos have audio. I didn't hook up the audio of this, so I think I'm just going to kind of turn up the volume on my computer, but I think you'll hear it okay. Hang on a second. So the Galant Lab, as well as being doing very good work, is also very media savvy and has made some YouTube videos that have gotten a couple of million hits, deservedly so because they're good. Okay, so let's see. Hopefully be able to hear this. Okay.
My name is Jack Gallant. I'm a professor of psychology and neuroscience here at UC Berkeley, and my lab works on vision. That's our main focus is on the visual system. And vision's a really important thing to work on because humans are very visual animals, and when humans have damage to their visual system, they're really incapacitated. And natural vision is a little like watching a movie. So essentially, we were trying to build a model that describes how your brain responds to movies. So if the external world is a certain kind of movie, we we want to be able to build a dictionary that will allow us to accurately predict what the brain activity would be that would be evoked by that movie. And then given some brain activity, we want to be able to reconstruct the movie that you saw.
That's the mirror.
So on the left here we have a movie that we showed as subject to the magnet. And on the right we have a brain. And you can see the brain activity is painted here on the side of the brain, but the brain's kind of crunched up in the skull, so it's hard to visualize. So we kind of inflate it, and then we flatten it out, and we can see a flat map of the visual cortex and the rest of the brain as the person watches the movie. The problem here for us is to translate between these movies that occur and this pattern of brain activity that occurs. And in the study we talked about in our paper, we just modeled this very back part of the brain, the early visual system, primary visual cortex. And that part of the brain responds to the little local features in the movie, little edges and colors and pieces of motion and texture. But this part of the brain doesn't know anything about what the objects are in the movie. First, we put people in the magnet for several hours, and we show them movies, and we build them all of their brain. And then in the second part, we put them back in the magnet, and we show them new movies, and we measure their brain activity, and we decode their brain activity in order to reconstruct the movie. So on the left here is the movie we actually showed people, and on the right is our reconstruction. When the movie that we showed has a fairly common object like a person, our reconstructions are actually fairly accurate. When the movie that we showed is something rarer, as you'll see in a second, like this abstract thing, then our reconstructions are coarser. I think you'll be able to use this kind of technology for communicating with people who can't communicate, like locked-in patients or people with neurological diseases. I think you could maybe use this in some forms of psychotherapy or dream analysis. I think there's a lot of potential clinical applications for this. When this model worked, we were very happy because it was really quite surprising that it would work at all. We're only taking snapshots of the brain every 1 or 2 seconds, and our reconstructions of movies happen at the movie rate. So that's much faster, and there's no reason one would think that this would work. And in fact, most of our colleagues just told us this would never work. So it was pretty surprising and satisfying when it actually worked.
Nice little "I told you so" at the end. But okay, so we're going to spend some time kind of unpacking how that worked. But let's watch another movie that they made, which is using this. So they've actually done really nice work. Using this approach, they then can ask quite interesting questions, such as, how would we represent particular categories of scene in the world? How would we attend to the world? How does that affect these maps? So before we start going into some details, we'll do some of that probably more also on Thursday. It's kind of interesting to see the kind of stuff they can do with it. So and this is another quite nicely made movie. This is like 5 minutes.
Hi, my name is Alex Huth. I'm going to tell you about a study I did here at UC Berkeley, along with Tsuji Nishimoto, Ann Vu, and Jack Gallens. When you look around the world, your brain is busily recognizing and categorizing everything that you see. So we presume that different categories of things are represented differently in the brain. But that doesn't mean that they're represented in completely different ways. Take cars and motorcycles, for example. They share a lot of features. They both have wheels, they both have headlights, they both go on roads. They fulfill pretty much the same functions. So you might think that they're represented pretty similarly in the brain. But how can we extend this idea to all the thousands of categories that we can see? One way would be to use a semantic space, where nearby categories are semantically similar, but distant categories are semantically very different.
This is like that
So
kind
what
of
does the brain
abstract
semantic
figure
space
we were
look
showing
like?
before, right?
Which of the thousands of categories that we can see are represented similarly in the brain and which aren't? To answer these questions, we put 5 human subjects in an MRI machine and recorded their brain activity while we showed them 2 hours of movie trailers, where we had labeled all the objects and actions that appeared during each second of the movies. Now, functional MRI measures blood flow in little rectangular areas called voxels. Our voxels are about 2 by 2 by 4 millimeters, and it takes about 30,000 of them to cover the entire cortex. So our next step was to determine how each category of object and action affected the blood flow in each voxel. We did this using regularized regression analysis. What we're left with is a model that tells us how each of the 30,000 voxels responds to each of the 1,705 categories in the movies. Then, we used principal components analysis, or PCA, to recover a semantic space from each subject's brain. PCA gives us a low-dimensional space, where categories that are represented similarly in the brain are close together, and categories that are represented very differently tend to be far apart. Interestingly, we found that the first 4 dimensions seem to be shared across our subjects. Now, of course, our true shared semantic space has more than 4 dimensions, but we're limited here by the size of our stimulus set and the resolution of fMRI. So what do the shared semantic dimensions look like? We're going to visualize them here using a graphical structure drawn from WordNet, which is a semantic taxonomy painstakingly hand-constructed by a team of linguists. Each category is shown as a node, and the links between categories show is-a relationships, like an athlete is a human. Verb categories, like communication verbs and movement verbs, are shown as separate trees. Here we're showing the first shared semantic dimension. It seems to distinguish things that move, like people, animals, and vehicles, from things that don't, like buildings in the sky. This is not really surprising, since we know that bright, fast things just tend to elicit more activity in visual cortex. The next 3 dimensions distinguish categories associated with people, categories associated with civilization, and biological categories. But instead of looking at each dimension separately, we can visualize all 3 at the same time. Now we've colored each node to show where it lies in each dimension. The red component of the color is set by the 2nd dimension, the green component is set by the 3rd, and the blue component is set by the 4th. So for example, the category that's high on the 2nd and 3rd dimensions but low on the 4th, like a mammal, will appear yellow. This makes it so categories that are represented similarly in the brain are assigned similar colors. Here we see that people and communication verbs are represented similarly, and animals are also not so different. Vehicles and buildings and movement verbs are also pretty similar to each other, but different from people and animals. This movie is showing the same data, but we've also positioned each node according to where it lies on the 2nd, 3rd, and 4th dimensions. So now we have a pretty good idea of what the brain semantic space looks like. We can use the semantic space to map out how different categories are represented in different parts of the brain. The first step is to take a high-resolution anatomical MRI of each subject's brain. Then we use this image to construct a 3D model of the cortical surface. Then we flatten out the cortical surface so that we can see the entire cortex at the same time. Now we can color in each voxel according to what part of the semantic space it's selected for. These colors are the same that we saw earlier. Green is for humans, yellow is for other animals, pink and violet are for vehicles, and dark blue is for buildings. What we see is a really complex pattern of semantic selectivity throughout higher visual cortex. These patterns are consistent with well-known semantically selective brain areas, like the fusiform face area and parahippocampal place area. But we also see semantic selectivity in much of the surrounding cortex. Now, if you find this as exciting as I do, you'll be itching to play with this data yourself. Well, you're in luck. We've made an online viewer where you can see the flattened and 3D brain maps. You can click on each voxel to see exactly which categories it represents. You can click on each category to see exactly how it's represented across the cortex. This viewer is available now at gallantlab.org/semanticmovies. So that's it. If you want to learn more, read the paper at Neuron. Thanks for listening.
So as you can tell, there's a lot more going on in that second movie than there was in the first one. So you know, we'll spend some time on Thursday, sort of unpacking a bit. Or maybe I'll just leave the lights down for now. Is that okay? Like, is that too? I'll spend a little bit of time unpacking, you know, some of what's going on. But let's start a bit by unpacking what's going on in the first movie, or at least some of it. And I think there's a couple, just as a kind of more general point. I think there's 2 reasons why the Gallant Lab work is just great work. One is because it's really interesting and just asks interesting questions about the brain. And the second is, these guys really know how to present their stuff well, okay? You could take that, I mean, you could take these kind of complicated sounding models and make it utterly indigestible, but they made beautiful, beautiful video demos which, you know, got 2 million hits on YouTube. And that's, in my opinion, that's not just advertising. I mean, it's partly advertising, and I'm sure that, you know, they were happy to get lots of hits on YouTube, but to actually, you know, to actually explain things that clearly, to actually make things that concrete, to actually really say, hey, I'm going to just show you what you did. Just watch this movie and bang, you know, you're going to get what it is. That's kind of really hard to do, and that's really, you know, that's really something we can all try and aspire to. And I think this is actually relevant for your class projects. So, especially if you haven't done too much of this kind of, pretend grant proposal type stuff before. Oh, by the way, I just wanted to say, one of you asked, does this grant proposal idea, have to be, an NIH format or an SF format? And the answer is, it doesn't really matter. The key thing is, the idea is to convey the ideas clearly and to convey, why someone should care. Okay? And I think that those videos are just kind of masterclasses in that, because they say, here's this problem that we faced. What's the question that we're trying to attack? Well, we're trying to, reconstruct images from visual cortex. How did we do it? Well, we built this model, okay? And they just kind of show the key aspects of the model. They don't go into all the nitty-gritty in the video. And for giving the big picture, that's fine. I mean, if you want all the key, all the details, read the supplemental information in the paper. And then, why should you care? Right? That's really important. So Jack Land, at the end of the first video, he said, well, this could be relevant for treating some conditions. He said this could actually be relevant for figuring out what's going on in dreams, which actually at the time maybe sounded like a really silly idea, but amazingly there's actually some people took the idea and ran with it, and this came out in Science like last year, I think, or 2 years ago, that actually basically tried to decode visual experiences that were perceived, people were having while they were dreaming, which is kind of, I didn't really expect anyone would be able to do that. And amazingly, they actually had some measures. I mean, this was not one of the readings I put in, but you can find this if you look for it. They had some measures which actually suggested that they weren't just kind of making this up, that they actually really were getting something right. So this I think that when you're kind of proposing your ideas, whether you're doing an analysis of new data and you're trying to explain why this is an interesting analysis to do, or when you're proposing an idea for an experiment, I would definitely recommend concentrating on these kind of big picture questions of, what question you're asking, how you're going to ask it, and why should someone care. And if you can make the first 3 sentences of your proposal be those things, which by the way would be really difficult to do, but if you can do that, then you'll be in great shape. And if you've got those first 3 sentences good, it kind of really doesn't matter what's in the following pages, really, because anything, if you go on and do this and you actually submit real grants, some of you I know have already done this, the person who reads your grant is going to be reading it probably at 3 in the morning, changing their baby's diaper in one hand and holding your grant in the other hand. They're going to be kind of tired and inattentive, and they'll just look through like a pile of 25 of these things. So you need to inject your ideas straight into their head in as seamless and undemanding a way as possible. So don't bury your key point in the middle of page 5 of paragraph 3, because they ain't going to notice it. And then you might say, well, they really ought to read my grant more carefully. Well, this is just the way people are built. So I would also recommend that, and that's actually, it's another of these things that's easier said than done. It's incredibly difficult to actually kind of boil down the key points of something to just 2 or 3 sentences. And in the second video, we'll spend some time unpacking this on Thursday, but we've already talked about this a little bit. In the second video, they were talking about projecting things to a low-dimensional space using PCA, primary principal components analysis. Okay? And they said, well, this obviously doesn't capture everything. And it doesn't. Whenever you take anything that's big and complicated, and you try and boil it down to just some very simple, low-dimensional representation, it's definitely not going to capture everything. So and it's kind of painful to just let stuff just go away in order to get that simplification. But usually it's totally worth it, because you're not going to capture everything when you try and inject these ideas into someone else's head either, but you're going to inject them a lot more successfully if you've kind of boiled them down a lot first. So anyway, so that was another reason why I wanted to show those videos. But so let's start to unpack a little bit. We'll just start this for a few minutes now. We'll do it more on Thursday. Let's start to unpack a little bit what some of the mechanics of this are. Okay, so the first stage is estimate a receptive field model for each voxel. So we already looked at what does the receptive field look like for an individual neuron. So their innovation was to say, well, hey, let's look at what the receptive field is for a voxel. Now, at first that might seem like a crazy idea because a voxel has millions and millions and millions of neurons in it. And these neurons are definitely not all doing the same thing. And they won't all have the same, even if it's a really tiny voxel by MRI standards, they will not all going to have the same orientation, selectivity. Remember in the Kamatani and Tong paper that we talked about, kind of part of the whole idea was that of why it would seem very, very difficult to decode orientation, that the orientation that someone was looking at using fMRI, you'd say, well, there's just no way you could do that because these orientation maps, remember these kind of woobly, colorful maps, I mean, they're pseudo-color, but lots and lots of orientations all swirling around, they're at a much, much finer scale than you could ever get even with the biggest MRI magnet. Okay, these are happening in a few hundreds of microns, the orientations are changing. But in a single voxel, which has lots and lots of different orientations in it, there might be a little bit of a preference for one orientation or another. And that was the idea that basically drove the success of the Kamatani and Tong paper. And so this Jack Galan work is very much building upon that. It says, okay, we know that a given voxel has millions of neurons in it. We know that those neurons are not all doing the same thing, but we're going to say, let's just see, after you put all of that big stew of neurons all together, if there's a particular preference that tends to emerge. Now, a very strong preference which is going to emerge is for a particular point in space, because that holds at a very coarse grain. So there's entire regions of the cortex which are actually responsive to particular parts of the visual field, especially around the fovea. So these very tiny bits here become very big chunks of cortex over here. It's called cortical magnification. So you can actually say with some confidence, even if I take some voxel from here, I don't know what orientation. There's going to be lots and lots of different visual orientations that those neurons are going to be tuned for, but they're all pretty much going to be tuned for this particular part of visual space. So when it comes to space, you're in good shape. But they said, hey, why not? Let's just see what happens if we look for tuning to orientation as well. And it wasn't a crazy idea because it worked for Kamatani and Tong. So, and not only do they look for orientation, but they look for spatial frequency as well. So remember, we've got these whole range of different spatial frequencies. If you ever do anything to do with computer vision, you'll definitely encounter this type of representation, which is often called a Gabor pyramid. And in fact, this is also the first stage of processing. When you open up your iPhoto or Picasso and it starts doing things like recognizing faces, it's almost certainly got something like this built into it. It was one of the early stages of processing. Okay, so for each given voxel, you say, we're going to show a whole bunch of pictures. So these are, I don't know if you can see this, but these are just a kind of like string of different images. So this is a picture of a couple of houses or something here and here. I don't know, that looks like a bit of a snake or something back there. Okay. So you show lots and lots of different images, and you say, for each of those images, let's see how much that voxel responded. Now, you could just kind of skip this whole kind of model stage in the middle and just say, well, why bother with that? If I find that, so suppose I know that the voxel responds to, this particular part of visual space here. You've got to make sure that people keep their eyes still in the center of the screen for this. Remember how they said they had like 5 subjects who they scanned for a couple of hours each? So these are not just, 5 random volunteer subjects. These are basically people from the lab who are very highly motivated to actually have, a couple of hours of, more than a couple of hours in some cases, of videos and still images shown to them, while they're keeping their eyes exactly fixed, exactly there the whole time, which is actually quite hard to do if you ever try doing that. So they basically kind of use themselves as volunteers, and that's actually quite common in these kind of studies because it's really hard to get someone to do that otherwise. And the worst thing, well, you can actually track people's eyes in the scanner, but if they start moving their eyes around, it's kind of, what are you going to do? Just like shout at them and say, please keep your eyes more still? It's not going to work. Okay. So, okay, so suppose you said, I'm going to skip all this stuff in the middle. Okay, I know that, say I'm recording through a voxel, and one thing I can be confident about is which part of visual space. If they're keeping their eyes stuck on the center, I can be confident that which part of visual space this voxel is responding to. So suppose I know that my voxel is responding to here. Okay, I can just say, okay, well, here's the response to this image. Well, that had this particular bit of image in it. Okay, and then what's the corresponding patch of the next image? And I can say, well, then I can just create the overall a picture of the overall response to that voxel by just, if it responds at intensity 0.4 to this image, and I've got 0.4 times this patch plus, 0.1 times the next patch, or whatever much you responded to that, why bother with all this stuff in the middle? Well, what do you think would happen if you actually just kind of took some weighted sum of the raw images, particular patch in the raw images? What would that end up probably looking like in the end after you have like hundreds and hundreds of images? I don't have a thought.
Would it be like if you just plot the distribution?
It would basically just look like a kind of moosh.
Oh yeah.
Yeah. It would look a little bit like it would look a little bit like if I took all of if I took everybody's fingerprints and I decided to make a class fingerprint and say, I'm going to average all your fingerprints together. You won't have anything that will tell you about any of the individual fingerprints. You'll just have this moosh. And so that's exactly what you would get here. If you just took a kind of response weighted average of the little bit of image patch, weighted by how much each voxel responded to each image, then everything would just end up responding kind of to this uniform gray mush, and it wouldn't tell you anything at all. So that's where kind of breaking down the images into their component parts, or one way of breaking them down into their component parts comes in handy. Okay? Because suppose you've got, I don't know if you can see this very much, but suppose you've got a voxel whose receptor field is kind of here, just happens to fall on this bit where there's like this black doorway next to a white wall. Okay, and suppose that makes the voxel fire a lot. Well, look, we actually have, in our kind of pyramid of differently oriented and differently positioned and different spatial frequency gabors, these little kind of grating patches, we happen to have some that actually happen to align, to be, vertical and aligned with this edge right here on this patch. Okay, so now we've got, instead of just being a little part of a patch of an image, we've got a component of our model, where the model is basically just the responses to all these different kind of filters, all of these different gabor patches, and now we can say, aha, this voxel really likes vertical edges right here, okay? Which is exactly what people do when they try and find the response of neurons in primary visual cortex, and you say, aha, okay, now we're getting somewhere, okay? Now we can say, now we actually have something that is actually more specific, not just to some kind of mushy average of lots of images together, but to the things that those images are built up out of. And they didn't just make this up. This is to a reasonable degree what primary visual cortex is doing. So they had a very, it was very reasonable to say, let's try this as our model. But remember how, we discussed when we were talking about Kamatani and Tom and Google and Wiesel. This is just a part of what visual cortex is doing. Like, there's nothing, these are all black and white images. I mean, they deal with color movies later. Okay, this is just black and white images in this original 2008 paper, and they're not looking at color at all. These are still images. They're not looking at movement at all. So in their later work, they kind of extended it. But even then, this is still just getting to a fraction of all the amazing things that the brain really is doing. But it's a pretty decent initial handhold. Okay? So you've got this whole set of different responses. And you say, okay, for each, so then you process the images. Okay, you say, to what degree did this image produce activation, or did it, fall upon a filter that was vertically oriented right here, or horizontally oriented right here, a particular spatial frequency? And then you say, given those components of the model, I'm going to weight them by how much the voxel responded. And so now you're starting to build up a picture of how the voxel responds, not just to these particular images, but to any image which might happen to have an edge at that particular part in that particular orientation. So you can start to see how this is a model that extends beyond the set of stimuli that they happen to train it on, but which could generalize to new images altogether. Anyway, okay, so that's just the beginning. Obviously, there's more to unpack on that, especially with what they started to do with the semantic space in the second video, and we'll talk more about that on Thursday. But this is kind of a nice approach that is definitely worth knowing about. So anyway, so to recap with the I'll send this out. Well, actually, this was already in an email. To recap with the project, and I will get back to you guys more quickly in the future iterations. Then the next question is really, try and figure out what do people know about this, what do people not know, and what might be interesting to look at, and try and think of the kind of write-up that you're going to be doing as really focusing on these three questions. What question am I going to ask? How am I going to ask it? And why should anyone care? Especially the why should anyone care bit. That's important. Another question is, well, how long should the whole thing be? If you can say what you want to say in a brief, in like 2-3 pages, that's fantastic. And in fact, actually, they're always cutting the length requirements on grants these days. If it takes you 35 pages to say what you want to say, then take 35 pages, but do not really expect that I or anyone else will be, as deeply attentive by page 27 as we were on page 1, because that's just how it works. I would say that the more compact and the more digestible you can make it, the better. And I think those Galant videos are just a nice illustration of, and that's kind of about as good as it gets for that kind of thing. So anyway, so that's the homework for this week. On Thursday we'll talk more about this Galant stuff. Thanks.
So you saw your email that you sent to me? So you said 11:30 on Friday?
That would work for you? Yeah. Okay, brilliant. And we can email you need to put it in all the in a very good shape. I mean, you're already doing stuff that is perfect. So you can just take, I would say, I think it's kind of fun. Just take the stuff that you're doing and say, oh, this is what we're doing. And we can kind of, to make it more cross-fertilizing, presumably there's, I think I'm doing something similar to say, well, maybe there's particular analysis that I would like to try, well, could you kind of, give me some advice on that? And
so I have some ideas, things I haven't done yet that I thought were cool to do. Trying to merge those ideas into more now.
I think it's easy, especially like the new method, to kind of say, take this question and then very compelling if you can say, there's a particular question that we really like to ask, and here's why we should ask it, and here's why you should care, and I'm telling you stuff you're going to say. And in fact, there's no way we can ask it unless we do this method, and this method will unlock the gates of knowledge and stuff like that. So if you can express it in those ways, or actually do it in those ways, then that would be, just
running the
okay, so we can email a bit before that. I'll even reply to that.
Cool, thanks.
Hi, how's it going?
Hey, it's going good. You did get back to my first proposal.
Yes.
Finally.
Yes.
Yeah, no,
No, but I'm sorry.
Actually,
It's
it was
very
before
bad.
that, so-
It's very bad that I was so slow about that. So now I have to remember. Please refresh my memory as to.
Yeah, so I'm looking at Aram or Aram.
Oh yeah.
I mean, there is, we can look at the response time, but I think that probably for our purposes, yeah, and I mean, like, JavaScript interpreters are pretty
Making
fast.
cognitive decisions, but then not.
In terms, so you don't have to worry too much.
Oh, like the
about
stop
JavaScript
signal.
in terms
Yeah.
of
Yeah.
So you mentioned a couple of things at the end of the email, but it seemed like you gave me a few directions to go
Okay.
over, so.
Okay. Let me.
No, so this,